Hi guys, happy Tuesday. So today is day 3. Today what I want to do and what was posted for you guys yesterday, you should have copied the last 2 pages back in front of your diagram packet. I want to go through those today, and then we're going to go through another little kind of review worksheet that you guys will be looking at. So if you guys will flip back to your diagram packet, I'm going to start off with a little RNA processing or the editing. So within our notes, we talked about some things with exons and introns, and I said they're kind of like when you're writing an English paper, and that first time you write an English paper, it's kind of your rough draft. That's what we see at our top strand right here. So within our DNA, there's a lot of segments or a lot of pieces of DNA that really don't code for anything. So through evolution, through invading viruses and how they put their DNA kind of mixes with ours over millions of years, we have some junk DNA. So those are called the introns. So what basically happens is as transcription happens, we're actually only going to be copying the exons, and they'll eventually are going to get all pasted together. So by the time that messenger RNA strand leaves the nuclear pores for the cytoplasm, it's just the exons, that good draft, kind of that final draft that we're looking at here. So all that junk material never really is going to leave the nucleus. So over here is kind of like that big picture version for you guys, going from DNA, transcribing it into messenger RNA. Remember, that process is transcription. The messenger RNA, that segment that we copied from DNA, is going to leave the nuclear pore, goes out into the cytoplasm. This is where translation occurs. You have your messenger RNA, you got your transfer RNA, and then you got ribosomal RNA putting together that ribosome down there. The ribosome is reading my messenger RNA. It is going from a 5' end to a 3' end, and it is stringing together amino acids. Transfer RNA with its anti-codon is going to match with my codon on my messenger RNA, bringing in the appropriate amino acid. They get strung together. Remember, these stringing together of amino acids is done through peptide bonds, and this segment of amino acids is going to be my protein or polypeptide. If you guys flip that page over, now we kind of want to see, okay, now we've got the protein. You guys are going to have 2 views of this. This is kind of the smaller view, but if we're starting at the top, once again, this is all within the nucleus. I have my DNA, double helix. It's being made into messenger RNA. Messenger RNA leaves the nucleus. It's going to be going to the endoplasmic reticulum. In this case, this is the rough endoplasmic reticulum. It's rough because it has ribosomes on it. Remember, ribosomes are where the protein is going to be constructed or put together. Once it is made, it's going to travel through the endoplasmic reticulum. Remember, they are the roadways. Eventually, we're going to make kind of this little transport vesicle, and it's going to make its way, that protein is going to make its way into the Golgi complex. Golgi complex, we said, is kind of like a place where it's going to be modified, it's going to be sorted, it's going to be packaged into a protein. I said, kind of think like the Amazon warehouse. You guys order something online, it gets the package, it puts new labels on it, ships it out to where it needs to go. So that's what's kind of happening with this Golgi complex here. Our Golgi body, Golgi apparatus, they're all called the same thing. That little package can then leave the cell membrane, if that's what we're going to be doing to it, or it can become a lysosome. So a lysosome contains digestive enzymes, so it's kind of like a little digestive sac, and it's able to break down old cell parts or something within the cell that we don't want there. The other thing, like I just said, that can happen to it is that that particular protein, once it gets packaged, it can be secreted out of the cell. If you remember that from when we did our transport section, that process is exocytosis. It's a form of active transport. It's going to need ATP for that to happen. The next page, you guys are going to see a larger version of this that's going to be kind of numbered for you, and that's what you guys should have copied. If you're trying to copy during this video, pause and start it again. I'm trying to keep the videos a little shorter because it takes a little longer to download them when I get above a certain number of minutes. So pause them if you need to. So here we are on the next page of it. They are numbered for you guys 1 through 8. Same thing within the particular diagram that you guys saw. So if we're starting down here in the corner, once again, DNA being made into messenger RNA. That's what step number 1 says. There's my process of transcription. It is taking place in the nucleus. Messenger RNA is going to leave. It moves to the cytoplasm. It goes to a ribosome. This ribosome can either be located on the endoplasmic reticulum, or we have these free-floating ribosomes out in the cytoplasm as well. And we'll talk about what happens to those particular things depending upon whether they're going to the endoplasmic reticulum to be made, or they're going to be made at a free-floating ribosome. So let's say we're here, and what's happening is that my amino acids are going to be strung together at that particular ribosome. That process is translation. The protein then enters and goes through the endoplasmic reticulum. That's my transport area. As it makes its way to the end of it, step number 5 here, and it's going to pinch off, make a little transport vesicle. We just saw that on the previous diagram. From there, that transport vesicle makes its way into the Golgi body or Golgi apparatus, gets packaged, it gets sorted, it figures out where it's going to go, and one of two things can happen. So let's say we're going to secrete it out of the cell. We make a little vesicle so that it can be secreted out of the cell. So let's say when we did the little cracker activity and you guys put cracker in your mouth to digest it down, salivary amylase, which is an enzyme, which is a special type of protein, was released from your salivary glands. This is how it got released. It got released through a little vesicle, exocytosis, into the mouth to break down your cracker. The second thing that can happen is it can be made into a lysosome. So this is number 8 here. Remember that lysosome contains digestive enzymes. It's kind of like the stomach of the cell, and it can either attach to a food vacuole if we need to break something down, or it can break down old cell parts with that particular one, and that's what you guys are seeing here. So I said there are 2 places that you're going to find ribosomes. Proteins that are made at free-floating ribosomes, which are all these little dots that you guys have within your cytoplasm. So proteins made specifically at the free-floating ribosomes, those are proteins that are going to be used more for inside the cell. So when we're making like those lysosomes and things to break down food vacuoles and whatnot. Proteins made at the rough endoplasmic reticulum, meaning the ribosomes located on that rough endoplasmic reticulum, they're the ones that eventually are going to kind of get secreted out of the cell. And that's what you guys are seeing here. These 2 pictures are just kind of breaking down a little bit more so you get a better view. So here's my endoplasmic reticulum, my Golgi body, and it's showing how it's making lysosomes. And that lysosome is either going to attach to a food vacuole and break it down, digest it, or maybe an old damaged cell part and wants to break it down. But that's showing the process of making those particular lysosomes. This one is making a vesicle that's going to eventually get shipped out. So it's showing you how at the ribosome, we got my little protein being made. In this case, it's a glycoprotein, how it's going to be pinched off and transported on from there. The last page that you guys have, we've talked about viruses. Well, viruses can go through this process as well. We talked about DNA viruses. We talked about RNA viruses. If we're looking at a typical DNA virus, and we said DNA viruses don't mutate as easily as our RNA viruses do. And the reason is because when that virus enters in, the DNA can take over our cell's DNA. Double-stranded, they can kind of just work its way into each other. From there, it can make its messenger RNA, it can make proteins, and then it can go through and make more viruses. The one that you guys see here, for example, could be influenza. And we know that influenza is an animal attacking virus. What it does is it enters in, it starts to take over our cell's DNA with its own DNA. It manipulates it to make more viral particles. And as that virus pushes out, he actually uses our cell membranes, which has little receptors or ID tags on them, so that he is a little bit more viewable or is able to be accepted by other cells, let's say, because they're going to be able to recognize this because it came from our particular cell, meaning those ID tags there. Your RNA viruses are a little bit different. So HIV, for instance, is an RNA virus. Because of this, it's going to be putting RNA into our cell, and that RNA has to kind of combine with the DNA. Well, it doesn't happen that easily. So what happens is that this virus is going to use an enzyme called reverse transcriptase, and that enzyme is going to allow this virus to work backwards. So it's going to use viral RNA to make viral DNA. So the viral RNA enters in. It's going to use the enzyme reverse transcriptase to make viral DNA. So it's kind of working backwards to do this. And because of that, it's going to make a lot more mistakes because it's not as easily done. Since it's working backwards, we'll have more base pairs that are going to be messed up along the way and constantly mutating that particular virus. That viral DNA that gets made from that reverse transcriptase becomes part of the host cell's DNA. Eventually, it can go through and make viral messenger RNA and eventually make protein from there. So here you guys see that little chain here. So we have RNA-V, which is my viral RNA, gets made into viral DNA. That's that reverse transcriptase that's going to be doing that. That viral DNA, we said, can become part of the host cell DNA, and it's going to take over the host cell DNA, cause it to make more viral messenger RNA, which causes it to make more protein, as it goes through this process of taking over the cell. So you see the same steps. You'll still see translation and transcription along the way. What you guys are going to be doing and kind of wrapping up with that, you guys should have this particular paper. I've attached it to this video as well, and you guys should be looking at this. I don't want you to go through and color them in. I'm not worried about you guys coloring little circles and finding my add needs and everything like that. But look at the questions that you have here. So each section has little questions, 5 or 6 questions along the way. So go through the front page. It is looking at, as I'm going from DNA to messenger RNA, it's going to ask you guys a few questions about it. As you guys go to the next page, there's translation. Same thing. Don't worry about coloring, but use this particular diagram to answer the questions. And then you have a whole overview. Here's my cell, here's my nucleus inside, and then my cytoplasm outside. Use that to answer the questions. On the back side, this is what I want to kind of give you guys a little help with. You guys are going to see a third type of codon chart. So this one's a little different than the wheel that you saw yesterday, as well as the box one that you guys saw yesterday. So what I'm looking at here is a few things. First off, this is my chain of amino acids that you guys have, and they're kind of listed up there, which means that these structures are going to be my transfer RNAs that you guys see. Remember, on the end of my transfer RNAs are my anticodons, my 3 base pairs. This strand is my messenger RNA. I would go through and kind of separate them out. Every 3 base pairs is a codon. Those every 3 base pairs are going to match up with my transfer RNAs, which are going to match up with my amino acids. This messenger RNA strand is complementary to my DNA. So every 3 bases in my DNA is a triplet. So if I know my amino acids, I should be able to figure out my messenger RNA. Once I have messenger RNA, I can determine my tRNA. These 3 match with the first one I have here. Remember, it's leaving as it leaves behind its amino acid. And then these messenger RNAs are also complementary to my DNA. But we know that some of these particular amino acids are coded for by more than one amino acid, or more than one codon. So I want you guys, if these are my amino acids down the bottom here, that's that strand that you see right there. They are alphabetical. Let's say we're looking at alanine. Let's look at our second one. Alanine is coded for by 1, 2, 3, 4 different codons. I want you guys to use the first row only if you're given more than one codon. So for instance, alanine, it says GCU. I don't want to stick that on transfer RNA. Yes, he's carrying in the amino acid, but it is my code on messenger RNA. So I'm going to put GCU there. That's what it's coding for. I skipped methionine. He's kind of easy. He'll go there for you guys. Then I want to go down and look at my second triplet. This is my second codon. And I know that GCU should be C. G goes with C. C goes with G. U goes with A. And then I can figure out my transfer RNA. G goes with C. C goes with G. U goes with A. So you guys should be able to figure out from utilizing these amino acids, my messenger RNA code, my transfer RNA anticodons, and then my DNA triplets down here. So I want you guys to complete that packet. That will be the first thing that we review on Wednesday. Once again, watch the videos. Pause them as you need to. Write down your questions. Put them under the comment section so that others are able to see them as well. And this will be our first little review to get you guys ready for your test on Friday. Okay, thanks.